Good morning to one and all. I am Dr. Tariq. Today we will discuss about microbial biotechnology practical. So the topic is biochemical test for bacterial identification. The test name is IMViC Test. So actually the 3 or 4 tests are the combined together. It's called a IMViC test. That means bacterial identification biochemical tests. So further detail, if you want, the below the link I will provide you. You can refer the future reference. Okay. Now IMViC test means IMViC is a series of tests that are useful in the identification of enteric bacteria. The tests included I means indole tests, M means methyl red tests, VI means Vogus proscore tests, and finally C means citrate test. So those 4 tests are called IMViC test. IMViC. Okay. Now we start about indole medium. Tryptone broth contains extracellular tryptophan. Okay, these are the principles of the indole medium. Okay, now the procedure of indole test means it contains inoculate tryptone broth with the young bacterial culture. Okay, so second one is incubate at 37 degree for 48 hours. Suppose we working in the IMViC test, definitely it is easily also the previously 2 days before it incubated that. Which one is the bacterial culture? Okay, then going to the result. The red ring at the top of the plate is positive, at the same time it's a E. coli. The colorless ring at the top negative, that means Staphylococcus aureus. Okay, so these are the 2 positive and negative bacterial result. The positive is E. coli and negative is Staphylococcus aureus. Okay, these are the easiest method of the indole test. Now the result shows that indole positive and negative. So these are the red color is a left side, it is a ring is formed. Okay, these are the tryptone. At the same time it is a positive result and negative in the colorless. Okay, so these are the, it's possible to it at the same time successfully it will come the result. Okay, next methyl red strain of E. coli or mixed acid fermenter. The degrade carbohydrates into acid N products such as lactic acid, acetic acid, succinic acid and formic acid. Okay, so methyl red contains the principles are there. Okay, the pH indicator at the same time necessary, so everything is present here. So the materials method, the media, MRVP media, peptone or glucose or K2HPO4 broth. It's easily available in our lab. Okay, so reagent means methylene red reagent, methylene red solution. Okay, it's present. The procedure is the incubate 5 ml of the broth with the young bacterial culture for 24 to 48 hours at 37 degree Celsius. add methyl red reagent into the broth. The final result, the color is positive, E. coli is present. Suppose faint yellow, it's negative, the bacteria name is Klebsiella. Okay, so these are the one of the MR tests, methyl red test. So this is the result. Negative is a yellowish color, positive is a methyl red. It is a red color. Okay, and the third test is the Vargas Procus test. That means V test. So these are the material VB test. Media peptone glucose K2HPO4. So this is the reagent. The procedure is incubate 5 ml of broth with young bacterial culture and at 24 hours to 48 hours at 37 degrees Celsius. Then add alpha naphthol to broth. Then add KOH, potassium hydroxide. Then the result is the red color broth positive, the Klebsiella, pale yellow is present, the E. coli is negative there. Okay, these are the another one of the most important. At the C test, that means citrate test. Simon citrate agar is used to determine an organic's ability to use citrate as a sole carbon sources. The carbon sources, most important one, the test. The citrate test is the principles are there. The pH change is induced by carbon dioxide, which is given of an byproduct of citrate neutralization. Okay, when it reacts the sodium and H2O in the agar, it rises the pH above 7.6. The organism must contain the enzyme citrate to degrade citrate. The citrate, the reaction is citrate, it's going to trace of, it gives to carbon dioxide, NaHCO and water. So blue color will change. That this is the procedure for materials methods required. Media, Simon citrate slant agar. We want a Simon citrate slant agar. Then how to proceed the practical? The slant is streaked only with the required bacteria. After that, 37 degree for 48 hours incubation period. Okay. So finally, citrate result, green color is negative citrate. That means the presence of E. coli. Then suppose the blue color is present, the citrate is a positive. The examples of the bacteria's name is Klebsiella. Okay, these are the positive blue color, negative is a green color. Okay, students, thanks to watching.